Okay, for gene regulation, what that is, like the name implies, it regulates or turns things on and off about how genes are expressed. So it will end up controlling and regulating what genes are expressed and what are not expressed. And when I talk about gene expression, the whole central dogma is that DNA, we haven't talked about this because we'll talk about it next week, but DNA starts off getting transcribed into mRNA. So I'll write that down. So DNA is transcribed into mRNA. This is transcription. And then mRNA is translated into amino acids. Amino acids. And we'll talk more about this next week, but I want you at least to know what we're talking about. So when I'm talking about gene expression, the gene itself is genetic material, so DNA. To express that into something actually having a more physical form, it will be amino acid. And amino acid change sequence will then become a what? A polypeptide chain, yes, into a protein. Very good. Okay, so gene regulation is regulated, and which ends up leading to the cells to differentiate. When we're talking about cells differentiation, we're talking about the cells actually being having some sort of becoming an end type of cell. So originally they start off as stem cells, and the stem cells can be any type of cells within the body. And then later on, as they differentiate, they are then quote-unquote destined to become one type of cell. For example, we have a pancreatic cell here, a white blood cell, and a nerve cell. And once they are on that pathway to become one of these cells, they have only a particular structure and a particular function for it. So they cannot be like the stem cells they were once before, where they could be any type of cell and have many different type of function. So even though all these cells have the exact same genes, they are limited to the type of cell that they are. So they have a particular structure and a particular function. And once again, gene expression, this is when the genes on the DNA are transcribed and then later translated into amino acid sequence to become a protein, which I just explained. And over here is just showing you this, where over here is the bundled version of DNA, which is the chromatin, and then the DNA itself. And over here, this is where transcription occurs. So over here we have the mRNA. And then from the mRNA, they get processed, which we'll talk about later. And then it gets transported out of the nuclear membrane here. So this is the nucleus. Into the cytoplasm, and that's where translation then occurs to make an amino acid sequence, right? Each one of these single dots is an amino acid. And then the amino acids later get activated, which we'll talk about later in this lecture, actually. Okay, so this particular slide, I'm going to just kind of introduce you to it. I do want you to know the terminology of it, but you don't have to understand it in full, okay? Because it's a little complex. So what a lac operon is just this particular gene within bacteria, and it codes for enzymes that break down lactose within the bacteria itself, okay? So they have different parts to it. The first part here is the promoter. It's colored in this light green shade over here. And the promoter is the regulator that either turns on or turns off this gene. Okay, so this is also where the RNA polymerase binds to. In this orange structure here, this is RNA polymerase. And what RNA polymerase does, it does that transcription that we talked about. And what is transcription? It converts DNA into mRNA, correct? So that's what this RNA polymerase does. Okay. So this is the promoter is where the RNA polymerase binds to make mRNA. The operator, which is this yellow portion of the DNA, this is between the promoter that we just talked about, this light green, and also the sequence of DNA where it ends up being transcribed. So this is the location where actually a potential repressor protein can bind to. And we'll talk about the repressor protein in this next click. So this repressor portion of protein here ends up binding to the operator. And when it binds to the operator, well, the RNA polymerase, which would have bound to the promoter sequence, will be prevented from transcribing anymore because there's something blocking it. It's this repressor protein. So this repressor helps regulates it as well by preventing the RNA polymerase from transcribing. And how this is ultimately regulated is through the presence or lack of lactose, the sugar. Remember what is this? Lac operon is a gene that makes what kind of enzymes? Enzymes that break down lactose, right? Okay, so if there is no lactose in place, does it want to make these enzymes? No, right? It will be a waste of energy to make these enzymes because if there's no lactose, there's no need for lactose enzymes to break it down. So as you can see, when there is a lack of lactose presence, this repressor protein will bind to the operator here. However, when lactose is present, it will bind to the repressor protein, inactivating the repressor, causing a conformational change. Conformational change, once again, is a shape change of the repressor protein. And then that's going to prevent the repressor protein from binding to the promoter region, the operator region. And so allowing the RNA polymerase to bind to the promoter to begin transcription. So, like I said, RNA polymerase can then therefore bind to the promoter, begin transcription to make mRNA. Okay. And later on, mRNA is translated into amino acid sequence. Okay, the whole purpose of that is so I could introduce you guys to, I'm going to reuse some of these terms when we're talking about the eukaryote. So that was an example of a prokaryotic, right? It happened in bacteria. So within eukaryotic cells or organisms, they will undergo all these different types of gene regulation. We'll go into each one of them one by one. Okay, and also, so first of all is DNA packaging. We already know that DNA in its natural state during interphase is in what form? It starts with a C. Chromatin. Right, very good. And naturally it's bound or packaged by DNA wrapping around histones, right? To make these nucleosomes. We talked about this last lecture. So when you increase the packaging of it, increasing it to become more condensed, RNA polymerase cannot access the condensed areas. And so that's why the packaging of the DNA itself prevents the gene expression from being expressed. This is actually helpful because there are certain things kind of like the X chromosome inactivation. So for females, they have 2 X chromosomes. In the cells that undergo mitosis, one of those X chromosomes will be inactivated. Since they already have 1 X chromosome, they don't necessarily need the other X chromosome to interfere with its transcription translation process. So instead, certain cells will have 1 X chromosome, just say from the dad's side, while the other cells next to it might be the maternal X chromosomes that are activated. So this is one way of regulating gene expression or DNA being transcribed and translated is whether or not they are packaged or not. So those that are packaged, like this X chromosome inactivation, it prevents it from being transcribed and translated. And over here it's just showing you the different areas where one X chromosome is inactivated. So here the orange chromosome is inactivated on this cell here. So that will make the other X chromosome active. So that X chromosome that is active has a gene for black fur. And these are at these kind of locations here where you see black fur on this cat here. However, the other inactivation is when they inactivate the one with the black fur gene. And here they have the orange fur gene being activated. So these location has cells that have the orange fur gene that's activated. Okay. There is a transcription regulation as well. This is using transcription factors. And these transcription factors are these proteins that you see here. And these proteins, they will bind at parts of the DNA itself, over here specifically to enhancer regions of this DNA. And it also allows the RNA polymerase to bind to the promoter. So these transcription factors are going to increase the amount of transcription occurring. Hence the name transcription factors. There are other proteins that are repressor proteins that do the exact opposite, right? So these proteins will then bind to different parts of the DNA called silencers. And when they bind to that, it's going to prevent RNA polymerase from transcribing the DNA sequence to mRNA. So it's the exact opposite of what the transcription factors do. Now that we're going onwards to talk about how RNA themselves undergo a processing sequence, there are different parts of the DNA. There are parts of the DNA where are exons, and here they're showing you the different types of exons here. And these are the parts of the DNA that will eventually be transcribed and then translated into amino acid sequence to become proteins later on. Other parts of the DNA, as shown over here, these portions of it are introns. So the introns are not expressed. These parts of the DNA do not end up becoming amino acids. So one way kind of remembering, I think of it, is like exons being expressed portions of the genes, while introns aren't. So here it's just showing you another, if this was a DNA sequence, you know, parts of it are introns that are not expressed while the exons are expressed. And then there's these enzymes that ends up cutting off the introns portions of it so that you end up having an mRNA sequence with only the exons over here. Okay, alternative splicing is when you have mRNA. So we have this sequence, right, over here, as shown over here, different parts of the DNA that gets transcribed, and now we have different parts, all the exons like linked together. However, alternative splicing, what it does is that even though these are transcribed and later will be translated, before they get translated, they can alternatively splice, meaning that this sequence doesn't have to be the end product of this DNA sequence that was originally there. So to form different amino acid sequences, it alternatively splice. And so over here are the different parts that are exons of the mRNA. So that's like one kind of combination, right, where the green ends up linking up with the yellow portions of the mRNA and get translated later to become a certain type of protein. However, here's another different type of sequence where you have the blue and the yellow together to form another different protein. The reason why we have so many different ways of forming several proteins, it's using the same DNA sequence though. So it's very efficient in forming different types of proteins even though we have the exact same DNA sequence. Okay. Micro-mRNAs. Micro-mRNAs, these are just small, very short-stranded, single-stranded RNA sequences. These will end up binding to parts of the mRNA that would have been translated into amino acid sequence proteins. However, these since it's single-stranded, it could complementary base pair with the mRNAs themselves. And when they do that, it prevents translation from occurring. If it prevents translation, we're not going to end up expressing that part of the gene that it originally coded for. So this is just another type of regulation. Okay, and then we have translation regulation as well. So prior to the mRNA from being translated, it is regulated as well. So for example, the erythrocytes, what are erythrocytes again? Red blood cells. These are prevented from making hemoglobin if there's not enough heme present. So if there's not enough of these heme molecules, remember the heme, those are the ones that have the iron ions inside of it to bind oxygen to it. And so if they don't have enough of these heme group, then the erythrocytes will not produce the hemoglobin itself, because it's kind of pointless to make something that you don't have all the complete parts to it. And so this is just an example of how translation is regulated. Before translating the sequence into hemoglobin, it needs to detect whether or not there's enough heme to produce this hemoglobin to begin with. Proteins are also activated as well. Usually this occurs after post-translational, so after translation occurs. So they start off as inactive protein, kind of like this insulin here, this proinsulin. This is when the insulin itself is not activated. And then after it gets cut over here, this part is missing, this entire sequence is missing. That's when it becomes the activated form of insulin. And do you guys remember one of the digestive enzymes maybe that was an inactive form but then became an active form later on? Anybody remember? Yeah, pancreatic, not amylase. Remember, there's pepsinogen that becomes pepsin. Yeah, no. That breaks down proteins. Pepsinogen was the precursor, so it was like a longer version of the inactive form of that enzyme. And then when it got broken down, it becomes pepsin. That's the active form. And then there was like trypsinogen becoming trypsin. Things. Okay, here's yet another way to regulate gene expression as well. It's signal transduction pathway. This is when there's a signal from another cell that will end up causing an influence of gene expression within this cell here. So here we have a signaling cell here that will send some kind of signaling molecule. What are some of the signaling molecules we discussed in our time together? One's from the nervous system. Any signaling from the neurons? They expel what kind of chemical? They're called neurotransmitters. Very good. And so the neurons themselves will exocytose those neurotransmitters, affecting some kind of response on the cell, the target cell that the neurotransmitters binds to. Hormones do the same thing too. So, so the signaling molecule from the signaling cells will bind to some receptor on the target cell itself. And once it binds to that receptor, it's going to cause a cascade effect that will occur within the cell itself. And that will end up activating some kind of transcription factor. And these transcription factors, remember what was a transcription factor? It's going to end up causing transcription to occur. So it will end up going into the nucleus itself, binding to the DNA. After it transcribes, it forms what again? mRNA. Yeah, very good. Okay, and that mRNA will then leave the nucleus, get translated into a protein, amino acid sequence. Okay, so that's that. And I don't know if you guys remember this huge thing I showed a while back, but once again, it's the exact same thing, but this time it's a hormone binds to its receptor, causing activation of all these proteins, converting ATP into cyclic AMP. That's going to cause a lot of different activation of different proteins. But one of the proteins that activates is this CREB. Once it's phosphorylated, it will go into the nucleus, as shown over here, bind to the CRE region of the DNA to end up causing transcription to occur. So all these different type of products from the transcriptions will occur. Plus there's other things as well that occurs that you don't need to know about, but it's just to show you how our cells is so complex and everything. And this is yet another one of these transcription factors that will bind to the DNA, causing some sort of gene expression to occur. Blah, blah, blah. Okay. So, homeonic genes in general. We talked about this in the last lecture, if you guys remember. Yeah, no? Maybe? No? These are the genes that will help cause the development of our organisms. Remember how these homeonic genes within vertebrates are repeated. In invertebrates, they have these homeonic genes, but in vertebrates, there's a repeated portion of it that causes us to form our backbone. For these homeonic genes that you see on this particular fly here, these homeonic genes, there are certain homeonic genes at the mid-body portion of the insect that will form legs. If there's some kind of mutation on the homeonic genes at the head portion of it, there's a potential that it will end up differentiating into legs instead of antennas in that particular case. And we talked about this before already when we talked about homeonic gene last time. It's just to show you that in the different types of species that we have very similar order in our homeonic gene. This order causes us to kind of deduce that we have some kind of common ancestry at some point in time. Okay, this one you don't have to know too well, but just know that it is a way that we end up looking at how what our genes are available and which are activated. And we use this process called DNA microarray. What they use is using a radioactive fluorescent on these single-stranded DNA. And these single-stranded DNA will bind to genes that are activated, so we'll actually cause it to end up binding to parts of it that are expressing, actively expressing that particular gene. And once it binds to it, then we know that okay, that gene is actually active, and this is what it codes for, basically. So this is the process. This process you don't have to know. So they end up using mRNA from the genes of a cell, and they use this process called reverse transcriptase. And this is found in some certain viruses and whatnot. HIV has a reverse transcriptase as well. And they end up making single-stranded DNA through this reverse transcriptase, making this complementary DNA. And then the complementary DNA with the fluorescence will bind to that part of it as well, and then that's what's going to end up lighting up, making it fluoresce. You end up rinsing the ones that are not bound together, so you don't have any interference. So you end up showing what's being transcribed within that cell. Okay, so yeah, don't worry about how it's done. Just know in general, this is just one way of having us see which genes are activated within what type of cells that they are from. Okay, plants are pretty neat because they can be cloned readily by just taking any cells that are already differentiated. When I say differentiated, do you guys remember what I mean by that? Remember how like stem cells, they're undifferentiated. So stem cells, they could be any type of cells. Well, depend on what type of stem cells. We'll talk about that later. But it could be any type of cell, quote unquote. But when they're differentiated, then they're destined, or quote unquote destined, to become one type of cell. So plants with differentiated cells, they can somehow be undifferentiated to form an identical plant. So this is actually used like for orchids. So they end up cutting or removing certain cells from the already mature orchid plant, culture it in a medium to cause it to undergo mitosis to form another orchid plant. And they use this for plants with also not only for orchids, but also for plants with very high fruit yields, or those that are resistant to diseases, to increase the amount of these type of good traits that we are looking for. Even seedless plants are undergoing this process too. And this kind of makes sense too, right? Because the seeds are used to actually, it's part of their reproductive cycle. And if they don't have seeds, they can't undergo reproduction. So the way to reproduce them is to have this cloning aspect of it. And then there's another way of basically regrowing parts from lost limbs. This part is called regeneration. And regeneration is used within salamanders. Whenever they lose a particular limb, they'll end up reversing the differentiation process to end up forming, once again, undifferentiated cells to become that leg again. And fragmentation, this we already did talk about. Remember fragmentation from the starfish or sea stars? Where even though you break off a portion of it, that portion of it will end up forming a new individual or a new organism from that part. Say again? Sorry? Oh. Yeah, that's right. You were disgusted by that. I remember now. For animals, they clone a little differently, though. There's this process called nuclear transplantation. And what they do is, first of all, they remove the nucleus, as shown over here, from an egg cell or from a zygote. And once they remove that nucleus, they end up inputting already adult nucleus into that cell. So an adult somatic cell is then injected into it. And if it survives, it can undergo mitosis. And then eventually, if we end up letting it implant it into a surrogate mother, it will end up becoming a new individual. Or we could end up growing into cell culture and then do different types of experiments on it to make it a specific type of cell line. So, yeah, a surrogate mother is needed if we're going to end up forming actual mammal or any kind of animals that we want to end up cloning, because especially for mammals, we need a surrogate mother to implant it into the uterus for that embryo to develop. Oh, that's Dolly, by the way. That's the first mammal that was cloned. There's different uses for clones. One is that when we clone agricultural animals, we clone them so that we could obtain desirable traits for them. There's also a purpose for it in research as well, so that we have the same type of control experiment. So if we're doing experiments on the rat, if the rats are all cloned from each other, then we know that there's no variation between the no genetic variation at least within each controlled rat, unless there's mutations and whatnot. It can also be used for medical purposes as well. So like these pigs here, they are used to basically harvest their organs so that those organs, when they get transplanted into a patient, are not rejected by that patient. Other potential uses for it is to help repopulate endangered species. So these are actually like species that are endangered that they were able to clone. And there's a possibility to possibly repopulate extinct species, kind of like in Jurassic Park. Hasn't been done quite yet, I don't think, but it is a potential. Huh? Possibly. I mean, we gotta find intact DNA for dinosaurs first of all that's stable enough. And this cloning process is a very hard process to undergo. And plus, there's always this ethical issue as well. Disadvantages of having clones. First of all, we always been talking about biodiversity, right? The more variation of something, the more chances that if some kind of change in the environment, they will be able to survive. When you clone something, you're having the exact same DNA. So if you have the exact same DNA, then they're not that diverse. And then if the environment changes, then you wipe out the entire, everyone that has that same genetic, all the clones. What's up, Elizabeth? Oh, I thought you were raising your hand, sorry. And plus, there's a chance that all these clones tend to, so far it seems like there is a relationship with clones and their health, how healthy they are. Whether they end up dying prematurely or they tend to have certain diseases as they grow older, that's also a disadvantage as well. That seems like there's some kind of relationship to these clones. There's also human cloning. So originally, we already talked about how mammals are very difficult to clone to begin with. So there's going to be a very small percentage that will develop normally. And we already talked about how there's this relationship where these clones tend to be not as healthy as they grow older. And always there's this ethics of cloning. And so this is always debatable whether or not we should do this or if it's right or wrong to do this. I'm not here to actually promote one side or another, but just to let you know that these things are like there, just informing you guys of what's happening in the scientific field. So all these are still like not concrete as to whether or not they are going to end up doing it or not, but there's been talks about having it done and whatnot. Okay, so there are different types of stem cells. There's embryonic stem cells, adult stem cells, and umbilical cord blood banking stem cells from doing that process. And we're going to start off with the embryonic stem cells first. So there's this process called therapeutic cloning. This is when you end up doing that nuclear transplant that we talked about, where you take out the nucleus from either an egg or a zygote and then implant it with the adult cell inside of it to produce embryonic stem cells. So embryonic stem cells themselves are undifferentiated cells. These are the cells that do not or can form into any type of cell it wants to within the body. So this is usually right from the get-go after a fertilized eggs begins. All those cells from then on are these embryonic stem cells. So depending on the different conditions that they are growing, they'll end up developing into specific cells. So they'll end up differentiating, whether it's a blood cell, a neuron, or muscle cells. It all depends on the different type of conditions that they are undergoing. Usually, there are certain things that so once they are differentiated, whenever they undergo mitosis, it's either to repair or to replace damaged cells. Once again, there's this ethical issue whether or not we should be using these embryonic stem cells for research or not, and the debate is whether or not what happens to this, whether or not the debate is basically whether or not to form these in the first place, because usually after you harvest these cells, then these cells the rest of the cells are going to be go to waste, and there are ethical issues about this, whether or not we should be doing that or not, type of thing. Adult stem cells, these have already been differentiated, partially at least. So for example, we have bone marrow cells in our bone, red bone marrow in particular. This has already differentiated a little bit, but it hasn't completely differentiated, because these bone marrow cells will end up becoming our different types of blood cells, whether it's leukocytes. Remember, what are leukocytes again? White blood cells, or erythrocytes as well. And whether or not this is a less ethical issue, that's still up to debate as well. Since these are adult stem cells, then we're not getting involved with the embryonic stem cells. However, it's still debatable as to whether or not this is ethical or not. Finally, we have blood that is taken from the umbilical cord or the placenta, and then this blood contains stem cells as well. They are less differentiated than adult stem cells, but however, they are more differentiated from the embryonic stem cells. They have been used usually to help babies with some kind of genetic risk. For example, this Krabs disease disorder is just usually a disorder or a disease to the baby's nervous system, and there are some that are helped by using this blood from the umbilical cord and placenta. However, it tends to be not as successful as the embryonic stem cells that we talked about earlier. So once again, there's always this debate as to whether or not to use it and whatnot. Okay, so moving on to a different topic about cancer. Oncogenes, these are mutated genes that will eventually cause cancer themselves. Usually these oncogenes are developed by viruses themselves. What happens is that these oncogenes will then produce abnormal growth factors. And what are growth factors again? They help cause transcription to occur to make proteins, right? So if these proteins are unregulated, then it will undergo mitosis several times without any kind of stopping them. And if they don't stop, they'll just keep on growing and growing. So they could either increase in number, or they become hyperactive too. So over here, this is just showing you the increase in number of these growth factors. If you increase the amount of growth factors, you're going to increase the amount of transcription and then later translation. If you make one that becomes hyperactive, you don't increase the number of growth factor, but that growth factor itself is constantly causing transcription to occur. Does that make sense? There's two ways of happening. One is to increase the amount of transcription factors or growth factors. If you increase the number of these growth factors, you're going to increase the amount of transcription and translation. If you don't increase the number of these growth factors, but just make these growth factor a lot more active, then it's going to increase the amount of transcription as well, which end up increasing translation. You end up
Getting cancer.
Yeah, you will end up getting cancer.
So why don't we stop it at the beginning?
It's because you end up having a mutated gene. And when you have a mutated gene, then you can't really go back and fix it unless you destroy the cell itself. Does that make sense? Because if you have a mutated gene, if it undergoes mitosis, it's going to carry that mutated gene onto its other cells, that the daughter cells, right? And those daughter cells will undergo mitosis and form. Yeah, it keeps on multiplying because this mutated gene is unregulated now. It doesn't have any regulation factors to prevent it from dividing. Exactly. So, for example, if we're talking about if it's found in the lungs, then it'll be lung cancer. If it's found in the throat, it'll be throat cancer, so on and so forth. The other way of cancer occurring is an abnormal tumor suppressor gene. And tumor suppressor genes, these are proteins that help prevent uncontrolled mitosis. So, however, if you have an abnormal of these tumor repressor gene, then it's going to not prevent tumors from forming. So tumors will end up forming. Does that make sense? There's a double negative. So tumor repressor gene, it prevents tumors from forming. Okay, it helps regulate mitosis. Or if you have an abnormal one that doesn't function anymore, right? If it doesn't function anymore, then it cannot repress that tumor anymore, and so it's going to undergo uncontrolled mitosis. Does that make sense? Yeah? Yeah. Mm-hmm. Bloodstream? Uh-huh.
So you go in and you do your test, okay, and it's negative, and then you go back, let's say, 3 months.
Uh-huh.
And the first signs, like, how are you going to be able to know it?
There are certain, like,
How soon do you know?
it depends on how fast it reproduces or how many are already there and stuff. Because there are certain ways to detect certain cancers. Like prostate cancer, there's certain antibodies that will bind to a particular cancer cell within it. And if you see an elevation in that amount, then you know that, okay, we have to be aware of it, kind of thing. Not to say that it already has that cancer already, but there's a potential for it to occur. And that's why they will end up taking cells at that location and then see whether or not those cells are these mutated cells that causes cancer or not. They could be normal cells, kind of thing. So to answer your question, it's like it really depends. All different types of scenarios. That's why it's so important to eat healthy, to exercise, to prevent these things from occurring. Of course, to have your own self-examination and just going to the doctor itself to get regular checkups to see. If you get regular checkups, then even though you do have the early stages of cancer, you can potentially still treat it, by either removing it or that type of thing. But if you don't get regular checkups, if you don't do these self-examination, so on and so forth, this cancer can spread not only, at that location, but it could spread to other location once it gets into your bloodstream. Just think of a cell, right? At that location, it's unregulated, so it keeps on growing and growing. But once it gets into your bloodstream, it could go anywhere now, and then attach to that location and keep on growing at that location as well. Okay, the progression of cancer. So we already talked about that mutation within the oncogene itself. Then that will end up causing abnormal amounts of mitosis. Then if there's a mutation in the tumor repressor gene, we also talked about that, that will prevent it from repressing that tumor. And then if you cause that to be inactivated, then you'll end up causing that tumor to grow. So that's if it's the tumor suppressor gene. However, if you cause a mutation within the activating growth factors, we already talked about that, it either becomes hyperactive or you produce a lot more of these growth factors that will end up causing the tumor. If it's a benign tumor, it will remain at that site. However, if there's a mutation on that benign tumor, then it could potentially become malignant. And then if it becomes malignant, it could potentially become metastasize. So over here, this is a defective non-functional protein. If it undergoes cell division without being controlled by anything, it will continue to reproduce multiples of it and becoming a tumor itself. It remains at the site because it's just multiplying at that location, right? It hasn't gotten into our blood vessels or anything like that. It hasn't grown enough to access other sources to go to other locations. But once it becomes malignant, then it starts going to other locations, neighboring locations. When it metastasizes, that's when it actually goes into the bloodstream or the lymphatic vessels, and then that's going to spread all over the place.
But not all tumors become.
Yes, not all tumors become malignant or metastasize. You're right. So if it's a benign tumor and it just stays at that location, then you're okay. It might cause, depending on where it's located at, right? If it's in the brain, even if it's a benign tumor, it's going to cause damage to those neighboring neurons because it's causing pressure against those neurons, which might damage it or cause it to die. So most mutations actually are not passed on to their children because they're mutations on the somatic cells. Somatic cells, remember, the body cells. However, there are cases where the gametes themselves are mutated, and if that occurs, then now we can pass that mutation to our children. So for example, this BRCA1, this is a cancer gene within the breast for one type of breast cancer, which can be passed on from mother to daughter. But most case scenarios, they're usually not passed down. Cancer is the second most leading cause of death. They are caused by things called carcinogens. Carcinogens are these chemicals or whatnot from the environment that causes cancer. One way of doing this is by UV light, and that's why it's so important to wear sunscreen to try to stay out of the sun, etc. The other one, very common, is using tobacco. So that usually causes lung cancer by smoking, or if you're using chewing tobacco, it ends up causing throat cancer potentially as well as mouth cancer.
If you're if you have lighter skin, you're more prone to skin cancer?
That's a good question. Yes, in some sense, because what happened, the reason why is because the melanocytes within your skin, they produce pigments to help. The darker the pigment, it prevents the UV light from penetrating to harm your nucleus and stuff like that. However, if you're lighter skin, you don't produce as many pigments, so you could increase your chances of damaging your nucleus of those skin cells. The thing about UV light and tobacco, it is very preventable, right? Like I said, using sunscreen, staying out of the sun, or just don't smoke or chew tobacco. There are different cancer prevention, having a high fiber, low fat diet, and using vitamins as well. Vitamins are usually called antioxidants, where they basically prevent cells from being oxidized that will cause mutations within your DNA. So that's it. Acknowledgement pictures. I do have another lecture though. Let me just pause it here.